Namaskar, my dear students. Again, we are with the third part of the topic photosynthesis. In the first and the second topic part of this chapter, I have discussed with you the process of photosynthesis in detail as it occurs in the C3 plants, where the primary acceptor of the carbon dioxide is ribulose biphosphate carboxylase, an enzyme which is the most abundant enzyme and is responsible for the process of fixation of carbon dioxide into its organic form. But it was seen later on that some plants have adapted to the dry tropical regions and have C4 pathway. As I have talked to you in my earlier lecture also, that the C4 pathway is different from the C3 pathway despite the fact that the starting product carbon dioxide and the end product glucose remains the same. In the C4 pathway, they are called as the C4 plants because carbon dioxide is first incorporated in a 4 carbon compound. Actually, through these plants have the C4 oxaloacetic acid as the first carbon dioxide fixation product. They use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Now photosynthesis takes place in these cells and they show a special type of anatomy to which we call Kranz anatomy. Now let us have a view as to how does this phenomena takes place. The primary acceptor of the carbon dioxide is 3 carbon molecule phosphoenol pyruvate and is present in the mesophyll cells. As you can see in this figure, the enzyme responsible for this fixation is phosphoenol pyruvate carboxylase, are simply called as PEPKs. It is important to see here that the mesophyll cells, they do not have ribosco enzyme, ribulose biphosphate carboxylase enzyme, they do not have. C4 acid, oxaloacetic acid is formed in the mesophyll cells. It then forms other 4 carbon compounds like malic acid or aspartic acid in the mesophyll cells itself, which are then transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release carbon dioxide and a 3 carbon compound. The 3 carbon molecule is transported back to the mesophyll cell where it is converted to phosphoenol pyruvate again. Thus, it completes the cycle. I will make a mention here that the carbon dioxide released in the bundle sheath now enters the C3 or the Calvin pathway, the pathway which is common to all plants, that is no longer different from the C3 plants. Actually, what is the difference? That in fact the fixation of the carbon dioxide is different and ribulose biphosphate carboxylase enzyme is not needed or not used here, as the fixation occurs in the mesophyll cell where it is not present, and what is present is the phosphoenol pyruvate carboxylase. The bundle sheath cells are rich in an enzyme ribosco, the ribulose biphosphate carboxylase oxygenase, but they lack PEP, phosphoenol pyruvate carboxylase. Thus, basic pathway that results in the formation of sugars, the Calvin pathway, is common to C3 and C4 plants. Now, let us summarize the general aspects of the C4 pathway. What happens? We can read it in the stepwise manner. Carbon dioxide enters the mesophyll cell and is converted into carbonic form, the bicarbonates. The bicarbonates react with PEP to form C4 acid, oxaloacetic acid. Then C4 plants, they again get converted into this 4 carbon compound is again converted into malate or aspartate, which is again transported to the bundle sheath cells. The 4 carbon compound is decarboxylated and carbon dioxide is released and is fixed by Rubisco and converted to carbohydrate, and it undergoes the Calvin cycle. C3 acid product, that is carbon 3 carbon acid product after decarboxylation, is transported back to the mesophyll cell to regenerate PEP. That is, the PEP phosphoenol pyruvate is regenerated definitely, and the after its conversion it enters into the Calvin cycle and rest of the aspects remain the same. Now let us see the special anatomy that the C4 plants they have. What is that anatomy? The particularly large cells around the bundle sheath of the C4 pathway plants are called as the bundle sheath plants, and the leaves which have such anatomy are called as the crans anatomy. Crans means wreath and is the reflection of the arrangement of the cell. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick walls which are impervious to gaseous exchange. That is, no gaseous exchange occurs since there are no intercellular spaces between the cells of the bundle sheath, as you can see in this figure. Um, if you cut the section of the C4 plants, then you will see that they show the crans anatomy and distribution of the mesophyll cell. Now this figure will show you the difference in the anatomy of the C3 and C4 plant. You can see in the C4 plants the waxy coating on the lower epidermis. Then the palisade, you can see it is present in the C3 plants, but no such distinguishable tissue is present in the C4 plant. Bundle sheath cells are present in the C4 plants, but such cells cannot be seen in the C3 plants. So these are the differences between the anatomy of the leaf of the C3 and the C4 plants. Now let us summarize the differences in the process of the photosynthesis of the C3 and C4 plants. The process which the C4 plant undergoes, that is called as the Hatch-Slack pathway, whereas the C3 pathway, they have the Calvin cycle. In C3 pathway, you see that there is only one acceptor, that is ribulose biphosphate. But C4 pathway, in mesophyll cells, the acceptor of the carbon dioxide is PEP, and in bundle sheath, it is again ribulose biphosphate. The first stable compound in C3 pathway is phosphoglyceric acid, hence called as the C3 pathway because it's a 3 carbon compound. But in case of C4 plants, it's the oxaloacetic acid, a 4 carbon compound. A very definite difference that crans anatomy is absent in the C3 plants, it is present in the C4 plants, as just we have discussed about. Now this is a very uh, remarkable difference, that optimum temperature range at which the process of photosynthesis takes place in C3 plants is 10 to 25 degrees centigrades, whereas the optimum temperature for C4 plants is 30 to 45 degree centigrades. Now this is in C3 plants because of lesser affinity of ribulose biphosphate for carbon dioxide. Concentration of carbon dioxide reduces to 50 ppm. What I mean to say by this is the ribulose biphosphate carboxylase enzyme has affinity for oxygen also, and when there is more oxygen present in the leaf, it tends to bind with the oxygen also, that we shall be discussing just now in the photorespiration. But because carbon dioxide has a strong affinity for phosphoenol pyruvate, concentration of carbon dioxide reduces and is less than 10 ppm. C3 plants are considered to be lesser efficient as compared to C4 plants in terms of photosynthesis, whereas the photosynthesis in C3 plants is 15 to 35 milligrams per diameter per decameter of the leaf area of the uh, plant per hour, whereas it is 40 to 80 of the leaf area per hour in case of C4 plants. Now another mechanism that we come across in the uh, plants, and that is the CAM photosynthesis, which is also called as the crustacean acid metabolism photosynthesis. And the plants which show this type of photosynthesis, they are called as the CAM plants belonging to family Crustaceae. That is, this process is an adaptation that are in arid conditions, in dry condition, in desert plants, because to prevent the transpiration, the stomata are closed during the day and open at night to reduce water loss. So what happens, that these are CAM plants, their stomata, they remain open at night, and because carbon dioxide is stored in the form of the acid before it could be used in the photosynthesis. Some examples of the CAM plants, they are cactus, orchids, agaves, etc. And this is the you can see that what happens in C3 plants, C4 plants and CAM plants. The first reactant is the carbon dioxide in all the three cases. The product in all the three cases is glucose, but because of their different habitat, different adaptations in which they exist, there are different pathway through which they finally enter the Calvin cycle to form the glucose. As you can see in the CAM plants, these are the desert plants and stomata are open only during the night time. So they use this carbon dioxide of their respiration and make the glucose and make the photosynthesis possible. So these are different pathways, out of which this C3 pathway, it occurs in many plants which are non-arid or non-desert plants, whereas the other phenomena C4 and CAM are also equally prevalent. Now the next we shall be discussing photorespiration. So my dear students, I would like to tell you that somewhere in the year 1920, Otto Warburg was a scientist who observed that presence of excess oxygen in the atmosphere inhibits the photosynthesis in green algae. He couldn't make out actually what happens, but later on this inhibition was found in green plants also and not only in the algae. And this inhibition of the photosynthesis by high level of oxygen was termed as Warburg effect and led to the discovery of photorespiration. The process of photorespiration was demonstrated by Decker and Teo in 1959 in C3 plants, because it occurs only in the C3 plants, and was very efficiently defined by Krutov in 1963 as an extra input of oxygen and extra release of carbon dioxide by the green plants. And what was the Warburg's effect? The inhibition of photosynthesis by high level of oxygen in the plants is termed as the Warburg's effect. However, the process of photorespiration, it does occur in the C3 plants, and this is the difference between the C3 plants, that it is not found in the C4 plants. Now in most of the C3 plants, what happens, that ribosco, that is the most abundant enzyme in the world, is characterized by the fact that its active site can bind to both carbon dioxide and oxygen. Hence, from its name you can identify. Rubusco has much greater affinity for carbon dioxide than oxygen. And in C3 plants, some oxygen binds to rubusco and hence carbon dioxide fixation is decreased. Here, the ribulose biphosphate carboxylase, instead of being converted to two carbon compound PGA, binds to oxygen to form one molecule of phosphoglycerate and phosphoglycolate in the pathway to which we call photorespiration. Now, photorespiration consumes oxygen and releases carbon dioxide without producing any energy or the sugar. Now let us have a look at the mechanism, that how does it takes place. The process of photorespiration, in fact, it takes place in the three organelles of the cell, and the three organelles which are involved, these are mitochondria, peroxisome and chloroplast. Now ribulose biphosphate carboxylase, the main enzyme of the Calvin cycle that fixes carbon dioxide, acts as a ribulose biphosphate oxygenase under low atmospheric concentration of carbon dioxide. How much low? About 1%, below 1%, and increased concentration of oxygen. In presence of high concentration of oxygen, the enzyme ribulose biphosphate oxygenase splits in a molecule of ribulose 1,5 biphosphate into one molecule each of 3 phosphoglyceric acid and 2 phosphoglycolic acid. The 2-phosphoglycolic acid loses its phosphate group in presence of enzyme phosphatase and is converted into glycolic acid. The glycolic acid, synthesized in the chloroplast as an early product of photosynthesis, is then transported to peroxisome, another organelle in the cell. The glycolic acid reacts with oxygen and oxidizes to glyoxylic acid and hydrogen peroxide with the help of enzyme glycolic acid oxidase. You can see this in this figure. Now, the hydrogen peroxide is destroyed by the enzyme catalase and glyoxylic acid is converted to amino acid glycine by transamination reaction. Glycine is now transported out of peroxisomes and it reaches mitochondria, where two molecules of glycine interact to form one molecule of serine. Now, amino acid serine returns to peroxisome where it is deaminated and finally glyceric acid is formed. The glyceric acid now enters the chloroplast where it is phosphorylated and forms phosphoglyceric acid, and then it enters into the normal C3 pathway. Now you have seen that several steps takes place during the process of photorespiration, but the process of photorespiration interferes with the successful functioning of C3 cycle and in C3 plants. In these plants, some oxygen does bind to the enzyme ribulose biphosphate carboxylase, and hence carbon dioxide fixation is decreased. Moreover, there is neither synthesis of sugar nor ATP. It results in the increase in the release in the carbon dioxide by utilizing the ATP produced by the photophosphorylation. Thus, photophosphorylation occurs in C3 plants, not in the C4 plant, and it would not be wrong to call it a wasteful process, as much as half of the photosynthetically fixed carbon dioxide may be lost into the atmosphere through this process of photorespiration. Sometimes it is called as the necessary evil in the C3 plants. Now the last we shall be discussing the factors affecting the process of photosynthesis. As understood, an understanding of the factors that affect the photosynthesis is very necessary after studying the whole pathway. Actually, the photosynthesis is under the influence of several factors, both internal inside the plant as well as external. The external factors would be the availability of sunlight, temperature, carbon dioxide concentration and water. And internal factor includes chlorophyll, the structure of the plant, the mesophyll cells, the chloroplast number, size, age, orientation of the leaves and the chloroplast, internal carbon dioxide concentration, and of course the chlorophyll, its amount as well as its nature. The plant or the internal factors are dependent on the genetic factors as well as the growth conditions of the plant, whereas the external factors are determined by the habitat in which it is occurring. But it would be very important, it is mandated to be aware of the law of limiting factors, because it determines as to whether the photosynthesis will occur or not under what conditions. When several factors affect any biochemical process, the law of limiting factors come into operation, and it was this law of limiting factor was given by Blackman in the year 1905. What this law states, it states that if a chemical process is affected by more than one factors, then its rate will be determined by the factor which is nearest to the minimal value. It is the factor which directly affects the process of its if its quantity is changed. What I mean to say by this, I will try to explain it once again, that despite the presence of a green leaf, optimum light, carbon dioxide condition, the plant may not be able to photosynthesize if the temperature is very low or very high. Then the temperature will become the limiting factor, the stopping factor. The leaf, if given the optimal temperature only, it will start photosynthesizing. This law always operates, because despite all other factors being optimal, if one factor is not there, that becomes the limiting factor for a process. Let us discuss these factors one by one. These are the light, water, carbon dioxide and temperature, which are very main things. The light, we need to distinguish between light quantity and light intensity and duration of exposure of light. While discussing light as a factor that affects the photosynthesis, there is a linear relationship between incident light and the carbon dioxide fixation rate. At a higher light intensity, the it gradually it increases as other factors may become limiting. And similarly, the light quantity is very important. Then the duration for which the plant is exposed to the light is also important. Increase in the incident light beyond a point causes the breakdown of the chlorophyll and decreases the photosynthesis. Next, we shall be discussing the carbon dioxide, which is a major limiting factor for the photosynthesis. No doubt the concentration of the carbon dioxide in the atmosphere is between 0.03 and 0.04%, but increase in the concentration up to 0.05% can cause an increase in the carbon dioxide fixation. And beyond this level, it becomes damaging. The C3 and C4 plants respond differently to carbon dioxide concentration at low light conditions. Neither group responds to high carbon dioxide condition. At high light intensity, both C3 and C4 plants show increase in the rate of photosynthesis. The fact that C3 plants respond to higher carbon dioxide concentration by showing increased rate of the photosynthesis leading to higher productivity has been used by some greenhouse crops such as tomatoes, bell pepper, etc., and they are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. Now next is the temperature. The dark reaction being enzymatic are temperature controlled because enzymes are very sensitive to the action of the to the temperature, though the light reaction also are also temperature sensitive. They are affected to much lesser extent. C4 plants respond to higher temperature and show higher rate of photosynthesis, while as C3 plants have a much lower temperature range for the optimal temperature. The temperature optimum for photosynthesis of different plants also depend on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to temperate climates. Next, let us talk about water. Even though the water is one of the reactants in the light reaction, the effect of water as a factor is more through its effect on plant rather than directly on the photosynthesis. Water stress causes the stomata to close and hence reducing carbon dioxide availability. Besides, water stress also makes the plants to wilt out, thus reducing the surface area of the leaves and thus metabolically active and also affects the metabolic activity of the plants. So water no doubt is directly needed by the photosynthesis process in a very small quantity, but indirectly it affects the physiology of the plants and hence the photosynthesis. Now this is the graphical representation of the factors that affect the photosynthesis. As you can see, the increase in the light intensity causes the increase in the rate of photosynthesis till it becomes constant. Similarly, the increase in the amount of carbon dioxide causes increase in the rate of photosynthesis, then it becomes constant. But increase in the temperature slowly increases the photosynthesis till it reaches the optimum value, then it drops at a very high temperature, as you can see in this graphical representation. So these are the various factors that affect the process of photosynthesis. That's all for the chapter of the photosynthesis. If you have any query, you can uh, leave your query on my email, or you can um, call me on my phone number that is at the end of this presentation. Till then, thank you, stay safe, and that's all for this process. Thank you.